Have you ever been heartbroken? Have you ever felt angry over something? Disappointed, maybe? During the summer, how do you feel hot? During the winter, how do you feel cold? All this is as a result of the nervous system. What if I told you that just watching this video, millions of cells in your brain and in your body are firing electrical, signals at a very terrifying pace? What will you think about that? Mind you, this is anatomy and this is physiology. And anatomy is what? Location, location, location. Physiology is what? Function, function, function. The nervous system is very complex, honestly. It's the foundation of our experience, our personality, who we are, our behavior. It's very similar to the endocrine system. Why? Because the endocrine system and the nervous system, they join hands together, all right? They are CNS twins. They join hands together to maintain the internal environment or internal coordination. But unlike the endocrine system, the nervous system employs electrochemical means to send signals or messages from cell to cell. Why the endocrine system communicates by means of chemical messengers, which is what? Hormones secreted directly into the blood, all right, to its target organ. So because of that, it's slower. But the nervous system is faster, extremely fast. What you need to know is that the sense organs, receives information about changes in your body and external environment and transmits these messages to the brain and the spinal cord. That's the sensory CNS, central nervous system. All right. Now the CNS processes this information, relates it to past experiences and what determines the appropriate response. And after that, the CNS issues commands to muscles and glands to carry out this information or this signal. This is exactly the task, the three basic steps the nervous system takes. This discourse I divided into three, all right? It's the other two will come in different videos. This particular one, we are discussing the nervous tissue under neurophysiology, all right? The nervous tissue specifically. We have the second video will be the spinal cord, spinal nerves, and the somatic reflexes. And the third video will be the brain and the cranial nerve. Oh, I have a fourth one, which is the autonomic and the nervous system and visceral reflexes properly, and as well as special senses. We are going to discuss a lot of questions, all right? So mind you, this discourse is as a recap, a review for your exams. It does not substitute for your lectures. It does not substitute for your lecture slides. So it's very, very important for you to read properly before watching these videos. You should watch it from beginning to ending, write the questions, try to create similar questions. The more questions you answer, the better for you for any exam, all right? If the potassium leak channels are suddenly blocked in resting neurons, what would happen to its resting membrane potential? Let me give you the option. It's okay. It would become more positive. It would become more negative. The RMP would be unaffected. It would be hyperpolarized. I want you to put down your answer in the comment section below, please. And mind you, you need to subscribe, you need to like, you need to also comment. Most importantly, you need to click the notification bell so that any time posting any video, any live stream, you'll be able to catch that information. If it's useful for you, you log in, all right? So that notification bell is extremely important, and your subscription is extremely important. Thank you. Which of the following is true regarding axons in the peripheral nervous system, PNS? A Schwann cell folds its plasma membrane around several axons rather. B, a Schwann cell wraps its plasma membrane around each individual axon as it does in the CNS. C, an oligodendrocyte cell wraps its plasma membrane around each axon as it does in the CNS. D, a satellite cell cluster around each axon to form a pseudomyelin sheet. E, astrocytes sends extensions called perivascular feet to wrap each axon. What is your answer? I want you to write the question, then put your answer beside it on the comment section so that I will tell you if you're correct or you're not. All right? I can also ask you which cells form the myelin sheets, in the PNS or in the CNS. Also, what is the primary on a neuron for receiving signals from other neurons? What's the primary site? Because there are different sites, but there is what? Primary. Or in which part of the neuron is the postsynaptic membrane usually found? These numerous other questions we are going to discuss. So I will encourage you to watch this video from beginning to the end, all right? Because a lot of questions will be coming at this beginning, in between, and also at the end. I hope you can see the slide I'm projecting right now, because the slide also contains some information that you need to know. So I'm discussing strictly from this slide. So I need you to pay more than usual attention to this discourse. Now, the nervous system division, you need to see the big picture. How is it divided? It's divided into the sensory division and the motor division. That's the Afferent and efferent. Afferent, sensory, afferent. So I touch someone, you feel something, you sensed it. That is afferent. This afferent carries information where? To the central nervous system. And what are the central nervous system? Mind you, the central nervous system is the brain and the spinal cord. We have the peripheral nervous system. The peripheral nervous system is every other thing that is not the brain and spinal cord. All right? So the sensory division carries information to the brain. Efferent information is arriving or away. All right? Now, the motor division carries information from the CNS backwards, exiting. Exiting. E for efferent. All right? Now, the question you need to understand is how does these things work? They can carry that information from the skeletal system or from the smooth and the cardiac, mind you. If that information is being carried from or to the skeletal system, it's called what? Somatic. If it is being carried from or to the skeletal, the smooth muscle and also the cardiac muscle, it is called what? It is called visceral. That's what you need to understand. So that brings us to the somatic motor division and the visceral motor division. So the visceral motor division is the one that controls smooth muscle, cardiac muscle, and glands. That's visceral. Most involuntary unconscious movements, all right? So it's very, very important that you know. And also there's something we call preganglionic and postganglionic. Example is how the heart moves, how the intestines move, how the glands are being secreted. All this is a visceral motor. So if, for example, I touch the skin, this is skeletal muscle, taking the information to the brain, it will be touch the skeletal muscle, that is what somatic, taking to the brain, away to the brain, that's afferent. So that is somatic sensory. Does it make sense? If it is coming to the skeletal muscle, it becomes what? Motor what? Sensory. If it is going to the heart, it becomes what? Motor visceral, because the heart is under the visceral section, all right? They can ask you some very important questions, like what is another name for the autonomic nervous system? The dash division carries signal to smooth muscle in the large intestine. These are some of the questions that they are going to ask you. So you need to be able to understand what the somatic is, what the visceral is, what the somatic and the visceral, the sensory and the efferent or the motor. All right? Extremely important that you know this. And another thing you need to understand is that the motor, the visceral motor, that's the one coming from the brain to the smooth and the cardiac muscle, is divided into two. One is the sympathetic nervous system, and another one is what? The parasympathetic division. Sympathetic is fight or flight. The parasympathetic is rest and digest. All right? This sympathetic prepares you for action. All right? Action. You know, releasing of norepinephrine at most stages. All right? What does it do? It increases the heart rate, dilates the pupil, inhibits digestion. So the sympathetic nervous system, it is activated in some organs, and you know it inhibits some organs. For example, in the heart, it makes the heart pump faster. The respiratory system goes faster, but it inhibits things like the nervous system, things like the urinary system, the digestive system. And this is exactly why you don't poo when you are running. You don't poo, you don't wee when you are running. All right. So very, very important for you to know this. Then the parasympathetic division, right, talks about relaxation, calm, rest. So your heart rate reduces, your respiration reduces. But it activates things like what? The digestive system, the urinary system. So when you are running for your life, something is happening, you are running for your life, you're under sympathetic control. Your heart is moving, beam, beam, beam. Your respiration is, your lungs is moving, all right? But your digestive system is inhibited, your urinary system is inhibited. So at that point, you are running for your life, you don't feel like pooing, you don't feel like peeing. Very, very important for you to understand this. So there's a question that they may ask you there is that, the dash tends to prepare the body for action. That's sympathetic. The dash, they can also ask you the sympathetic nervous system, what is its effect to urinary system? What is its effect to digestive system? All right, they are trying to test you. So this is receptors and effectors. You need to understand the receptors and effectors. The receptors is the stimulus detectors. All right, they are located in the sense organs. They detect the environmental changes, that's the stimuli, and they convert these stimuli to electrical signals and send them straight to the brain. In the brain, there's something we call what? Interneurons that explains the entire information from the receptors. Now, it will not send it out, all right? When it gets to the effectors are the ones that, the response generators, kinda, all right? They can be glands, they can be muscles, all right? And the muscles, they can be cardiac muscles, smooth muscle, or skeletal muscle. So if the information comes to my hand, my smooth muscle here for me to contract, I'll contract. And this smooth muscle, this skeletal muscle rather, becomes what? it becomes what? An effector. So you need to understand what are receptors and what are what? Effectors. Mind you, they can ask you a question. Which of the following are effectors? All right. They'll give you different options. Sometimes the answers can be glands. Sometimes the answers can be muscles. Sometimes the answers can be anything. But you need to be able to understand it. That, okay, this is something that is what? Giving out that response. This is something that is what? Giving out that response. So one detects response, another gives it out. The one that detects is what? The receptor. The one that gives it out is what? The effector. Okay. Now, there are functional classes of neuron. We define neuron into functional and structural. The functional, we have the sensory neurons, the motor neurons, and the interneurons. I've already mentioned a lot about this. Sensory neurons are the detectors, the stimuli detectors. They carry signals to the CNS. The motor, the ones, the efferents, the one that bring the information back. Interneurons, the ones that do what? That interprets. And these interneurons make up about 90% of all the neurons in the nervous system. And they are found specifically in the brain and also in the spinal cord. All right? Now, they can ask you a question. Some dash neurons specialize to detect stimuli, whereas dash neurons send signals to the effectors of the nervous system. You have to understand. What are they asking? They ask which one is representing the efferents, which one is representing the efferent. They can also put that option in such a way that which one is the sensory, which one is the motor. So you need to be able to understand what the question is talking about for you to be able to attempt it. Now, remember I mentioned that we have functional classification. Now, this is structural classification. When you are talking about structural classification, you are talking about things like what? You are talking about things like the multipolar, the neuron being multipolar neurons, bipolar neurons, unipolar neurons, and anazonic neurons. When you are talking about multipolar, what are you talking about? You are talking about the one. Look at this diagram. You see, this is a multi. This is a multi. You are talking about multiple dendrites with what? One axon. This is one axon. This is multiple dendrites. 1, 2, 3, 4, 5. This is multiple dendrites. This is one axon. That is what? Multipolar. And another thing you need to know about multipolar is that multipolar is the most common. They will ask you, which one is the most common? 99% of the neurons in the brain is what? Multipolar. Now we have the bipolar. Dipolar is what? One axon. This is the axon. This is one dendrite. One axon, one dendrite. Where is it found? Usually in the retina and in the olfactory epithelium. Very, very important for you to know. Now we have what? Unipolar. You are unipolar. This is an example of a unipolar. If you can see the slide, this is an example of unipolar. Unipolar, single process that splits, all right? It's just like T. Look at it, T, all right? Now, it's found in neurons, in PNS, all right? It has a pseudo-unipolar development. So, this is unipolar. Now, we have anazonic neurons. Anazonic neurons, no distinct axon. No distinct what? Axon. This is anazonal. You see, everything is dendrite. You can't see axon there. You understand? We also have pseudo-unipolar here as well. So, this is pseudo-unipolar, this is unipolar, this is anazonic, this is bipolar, this is multipolar. Where your question always comes out from is multipolar and what? Bipolar. They can ask you what is the common type of what? Neuron. The most common type is always multipolar. And they can ask you other questions connected to it. All right. Now, in terms of nerve organization, all right, from fiber to organ, what do we have? Nerve fiber is equals to single axon. An axon is a nerve fiber. Now we have bundles of what? Many nerve fibers. That's bundles of axons called what? Nerve. All right. Bundles of axons called what? Nerve. So nerve organs, not tissues, not cells. Nerve are what? Organs. But nerve fiber is what? A single axon, which is what? A cell. You need to bear that in mind. Extremely important. So when you are looking at nerve fiber or an axon, you should be thinking of what do we have in a cell. It contains endomerion, perineuron, and what? Epineuron. Epi is always the outermost. This is epineuron. All right. Perineuron is the bundles of axons that are joined together to form what? Fascicles. Remember, if you look at my muscle fiber video or muscle tissue video, we also discussed the same thing. We have muscle fiber, all right, which is the cell. Group of muscle fiber is known as what? Fascicles. Group of fascicles now forms what? The muscle tissue itself. So the same thing with this guy here, all right? Bundles of axons are what? The cells. Bundles of the cells are called what? Fascicles, and they join together to form what? Nerve, which is what? The organ. And each axon or each cell is surrounded by what? An endoneuron. They contain things. So a nerve structure properly contains what? An axon, blood vessels, and what? Connective tissue. Very important. Now, when you are looking at a neuron properly, there are three main regions. We have the input region, we have the integration region, and what? We have the output region. when you're talking about the input region, I want you to see me as a neuron, all right? If you see me as a neuron, you see my long hair? You see my long hair? This my long hair is what? Is the dendrite. This my face is the cell body, all right? This neck is the axon hillock. And this, my body downwards to my leg, is what? Is the output region. That's the axon and its terminals, all right? Extremely important that you can think of it this direction. So if you think of it this direction, they will ask you which part received the information. The information is usually, majorly, primarily received by the dendrites and also the cell body. Very important. And where integration happens in is where the axon hillock. That's where it decides, should I fire or should I not fire? Should I form action potential or should I not? All right. Now we have the downwards, which is what? The axon itself and the terminals. These are the ones that transmit information from this particular neuron to another neuron. All right. Even from this particular neuron to a muscle. That's why we have a neuronal transmission and also neuromuscular transmission. Extremely important. Now, the structural part of a neuron that you need to know is where the dendrites, we discussed it, the dendrites, the cell body, the axon hillock, and the axon itself. Then downwards, you talk about the collaterals, that's the 5 branches, and also the terminals. Extremely important. So it has a very specialized information for everything. Now let's talk about the cell body. It has specific functions. It's very specialized environment, all right? It has specific function. And what is it? It's that the cell body, remember my face. I need to be looking at my face. Okay, so the cell body, it contains the nucleus. It houses the nucleus. So it's genetic what? Control, all right? So most activities happen there. Remember, in the nucleus, we have the DNA function, blah, blah, blah. All those activities happen there. So it's the site of metabolic and regulatory what? Functions. Protein synthesis, ribosomes, rough ER, all of them are where? In the cell body. So it makes sense. Because remember, the synthesis, protein synthesis, DNA replication, and everything starts where? If you look at my genetics video, you learn this, all right? And before I continue, please, if you've not subscribed to the channel, can you go and subscribe now? I want you to subscribe. I want you to make comments. Even if you don't know what to say, just say anything. Just say thank you, all right? So make comments and like. Most importantly, put on the notification button so that you'll be able to, know when I am going on live and also when I bring other part of this neuro, all right? And send it to your friends, the link to your friends, all right? Thank you so much. Now, as I continue with the soma, as I said initially, is the site of most metabolic activities, all right? Protein synthesis happens there as well. Ribosomes are there. Remember, the protein synthesis happens in the kitchen. That's in the ribosome. Ribosome is found where? Either free or in the rough endoplasmic reticulum. Now, there is mitochondria because it needs it for energy production, all right? So it maintains the entire health of the neuron. It contains Golgi complex for packaging of the produced proteins, cytoskeleton for structure and for support, and also nuclear bodies, which are protein manufacturing things found where? In the rough endoplasmic reticulum. If there is a problem with the cell body, the entire cell dies because how can it produce? How can it create anything? All right, which is different from the axon. You understand? If the axon is damaged, it can actually, be fixed. It can be fixed. The astrocytes are there. Even the microglia is also there to support. All right, and these neurons can be found in the CNS or where? In the PNS. So they can ask you a question. Most metabolic and regulatory functions in a neuron happen where? You should be able to understand the answer, which is the cell body. All right. Now let's talk about the second one, right? Which are the receivers. That's the dendrites. You see, I told you it's the primary listing for receiving the dendrites. All right. So the dendrites are also highly specialized, like the cell body. They are branched extensions, just like a tree. All right. They contain receptors for neurotransmitters. Neurotransmitters coming from another neuron. They have the receptors for it. All right. So that's the idea. They are the primary sites for reception. All right. They are covered with spines, which increases their connection. They are the primary sites for receiving information, as I mentioned initially. Postsynaptic membrane usually is found there, and they generate local potentials. With time, you understand the difference between local potential and action potential. All right. Local potential graded, action potential not graded. Local potential decremental, possibly. Action potential cannot be decremental. Stuff like that. We'll still discuss it later. Now, the dendrites is very, very important for signal processing. Extremely important. All right. There are some questions that they'll be asking you on it. Also, remember, it has a specific ability which we call plasticity. That means it can grow or retract based on what activity. So it can learn. It can learn. That's the idea of dendrites. Some of the questions you may be getting is things like, what is the primary site on a neuron for receiving signals from other neurons? In which part of a neuron is the postsynaptic membrane usually found? Where do the most local potentials form in a neuron? Remember, local potential, it has to be where? In the dendrites. Remember, if they tell you postsynaptic membrane usually found, it is found where? In the dendrites. And what if they ask you what is the primary site for receiving neuron, for receiving signals from other neurons? It is the dendrites. Dendrites. So I need you to be able to understand this well so that you don't fail any question on it. All right. Now let's talk about the neck. Remember, we've described the dendrites, we've described the cell body. Let's talk about the neck. Now, how do you internalize this thing? The dendrites receive this information, then the information is processed in the face. All right, that's the cell body. Now, after that, everything comes to the neck. What is the function of the neck? That is the axon hillock. It decides, should this be a local potential or should this be an action potential? So the only place that could, authoritatively push out action potential is what? Axon hillock. Then we ask you that question. So what does the axon hillock do? It submits all the inputs from the dendrites, from the cell body, integrates them together. And if it is greater than the threshold, action potential fires. If it is less than the threshold, no action potential. It's as simple as that. All right? It's as simple as that. So we call it what? Trigger zone. So it integrates. Those local potentials could be in form of EPSPs, that's the accessory postsynaptic potentials, inhibitory postsynaptic potentials, right? It submits them together, you understand, until they are able to what? Fire. So the lowest threshold for firing is found where? In the axon hillock. That's what we decide. Immediately that threshold is crossed, it fires the action potential down the axon, pushes out down the axon. If not, it won't. It's just be local potential. So they may ask you a question: where do most local potentials form in a neuron? Where do most local potentials form in a neuron? You have to be thinking of what? Axon hillock. Now we are done with the neck. That's the hillock. Let's talk about downwards, which is what? The axon. The structure of the axon. You need to be able to understand the structure, the function, and the terminals. All right. Remember, structure, that is anatomy. Anatomy is what? Structure, or location. Physiology is what? Function. All right. So it's very important that you be able to understand these two things properly, because this is important. A lot of questions comes out from me, and I'll show you. A single long fiber, which is the axon, just long. Sometimes it has collaterals. That's what we call collaterals. That's branches, multiple branches. All right. Uniform diameter. Some of them are covered, or majority of them are covered by what? Myelin sheath. Myelin sheath helps to increase the rate of conduction. All right. Now, its function is just to conduct action potential. So that action potential that is produced in the axon hillock is conducted through the axon downwards. And another thing you need to know is that action potential are not decremental. So since axon only carries action potential, then the transmission is not decremental. It doesn't get weak. It must go strong. What is decremental? Local potential. And local potential is not shoot out by the axon hillock. If the axon hillock forms local potential, it keeps it within. It doesn't send it through the axon. It's as simple as that, because it's not up to threshold. Now, the axon itself demonstrates what we call all or none law. If it doesn't get the threshold, it won't shoot. It's as simple as that. It shoots when it gets to what? Threshold. Now, if you look at the structure of the axon, at the end part, you see what? Swollen end is that form what? Synapses. You see that big end, it forms synapses, and that is where, for the presynaptic, all right, that's what produces what? Neurotransmitters. Those neurotransmitters are stored in the synaptic vesicle. So when it wants to transmit neurotransmitters, calcium enters inside, breaks up the vesicles, and pushes out the neurotransmitters through what? Through exocytosis. And that's how action potential is transmitted from the presynaptic ward to the postsynaptic, because that's where synapses happen. I'll show you. That synapses can be with dendrites. It can be, this axon can be synapsing with a neuron, another neuron dendrite, or another neuron axon, or another neuron what? Soma. It can be anything, all right? And if it is synapsing with that axon, if it's synapsing with another neuron dendrite, it's called what? Exodendritic. If it's synapsing with another neuron axon, it's called what? Exoaxonic. If it is synapsing with another neuron what? Body, it's called what? Exosomatic. You need to know these terminologies. Extremely important. Now, let's talk about glial cells. What are glial cells? Listen, any system you are doing in anatomy and physiology, you should always know that it has what? Support. It's just like the digestive system, for example. The liver is just a digestive organ. It just supports. So the same thing in the neurons, in the brain, we have what? Supporting cells. In the nervous system, we have supporting cells, all right? And these supporting cells, we call them what? Glial cells. Extremely important, glial cells. And glial cells, they are so much more they outnumber the neurons by 10 to 1. 10 to 1. And you know, we have all to 1 trillion neurons. So that means glial cells should be almost 10 trillion. And another one thing you need to know about these glial cells is that glial cells, they do not generate or conduct action potential. They have no business action potential. They're actually mitotically active, unlike the neurons. The neurons are not mitotically active. So a neuron that is dead is dead. So neuroglia, nerve glue, it helps to hold the nerve. It has other general functions. All right, we have the physical support and protection, maintenance of chemical environments, they form myelin sheath, they supply nutrients, they remove debris, and they guide neuronal development. It depends on which neuron are we talking about. There are two populations of neurons. We have 4 neuroglia, rather. We have 4 glial cells in the CNS, and we have 2 glial cells in the PNS. Extremely important for you to know this. 4 in the CNS. So you need to know which one is in the CNS, one is where in the PNS. So let's talk about the CNS glial cells. We have the oligodendrocytes, we have the astrocytes, we have the microglia, we have the ependymal cells. You need to know what each and every one of these produces. The oligodendrocytes, specifically, they produce what? Myelin sheath in the spinal cord and the brain. Spinal cord and the brain. Now, in the PNS, what serves exactly the same function is the Schwann cells. But one of the major differences between the oligodendrocytes and the Schwann cells is that apart from the oligodendrocytes being only in the CNS, all right, one oligodendrocyte can form myelin sheath for multiple exons, almost up to 50 exons. Just one oligodendrocyte. But Schwann cell, one Schwann cell, one exon. One Schwann cell, one exon. So the oligodendrocyte wraps their membrane around the exon segments. Extremely important. But if they ask you, what does he do? Myelin sheath. Check it out. Now you have I'll talk about the microglia. Microglia, they are immune cells of the CNS. All right. What do? They help to, they are janitors. Janitors, you know, just like the lymphatic system. They mop up all debris, pathogens. These microglia are produced by what? By the monocytes. That's the white blood cells. So that's why it's very active during injury or disease. Extremely important. So we have the oligodendrocytes, myelination, microglia, they are the soldiers, the military or the janitors. All right. Then we have what? Ependymal cells. Ependymal. What does it remind you of? Epithelium, covering. Okay. So ependymal cells, they cover the brain ventricles, the pathways. They are the road network of the brain. Extremely important. And what do? They help the cerebrospinal fluid to move. They secrete and circulate. But mind you, what produces this cerebrospinal fluid specifically is, you know, the current plex. So this current plex, this is just, you know, network of blood capillaries. So with the ependymal cells, they produce what? CSF. And the ependymal cells circulates the CSF, all right, around the brain. So whenever we're talking about CSF, talk about ependymal cells. Whenever we're talking about immune system or protection or debris mopping up or injury, talk about microglia. Whenever we're talking about myelin sheath, talk about what? Oligodendrocytes. All right. Now, astrocytes, the most abundant. They will ask you which one is the most abundant. That's astrocytes. They are most abundant glial cells, star-shaped with many processes. They form blood-brain barrier. Blood-brain barrier. BBB. Blood-brain barrier, astrocytes. I'm repeating it again. Blood-brain barrier, astrocytes. Oligodendrocytes, myelin sheaths. Microglia, immune system. Ependymal cells, CSF. Check it out. Now, in the PNS, these are the 2 in the PNS. They ask you which 2 are found in the PNS. We have the Schwann cells and we have the satellite cells. I've told you about the Schwann cells. It works like the oligodendrocytes. What it does is it forms the myelin sheaths, all right, in the PNS. And as I told you, it's 1-to-1. Unlike if you look back, you see the oligodendrocytes. What did I tell you about oligodendrocytes? 1 to multiple, up to 50. But Schwann cell is what? 1-to-1 relationship. You understand? It forms the neurolemma, which is the outer nucleated layer, all right? Essential for PNS nerve regeneration. Very, very important. Then the satellite cells, they surround the entire cell bodies in the PNS. Mind you, PNS. We're talking about Schwann cells. So S and S is found in where? The PSS 51. So PNS, S, Schwann cell, another S, satellite cell. So you have to remember this thing and what they do. The PNS, they have better regeneration capacity, right, than the CNS. Why? Because of these Schwann cells, which is very essential for PNS regeneration. So the Schwann cells, they guide regeneration of exons. They will ask you in the exam. For here, the sample question, which glial cell is found wrapped around the exons in the PNS? What is it, PNS? Please, don't mistake it for what? For CNS. CNS is oligodendrocytes. PNS is what? Schwann cell. So this is a comparison of both Schwann cell and oligodendrocytes. You see the location? PNS. Oligodendrocyte location? CNS. All right? Schwann cell wraps about 1 exon per cell. Oligodendrocytes, multiple exons. one man marrying a lot of wives. Schwann cells forms neurolemma, that's outer layer with nucleus. But oligodendrocytes, no neurolemma formation. Supports regeneration effectively. So Schwann cells supports regeneration in the PNS, limited regeneration in the CNS. All right. So these are the differences. All right. But both of them form myelin sheets, which are lipid insulations. All right. And this myelin sheet helps to speed up action potential. In between the myelin sheets, you see what the nodes of Ranvier. That's gaps between the segments. Questions that they can ask you, you can see you are questions like, where are exons surrounded by Schwann cells found? You will know now. PNS. Which of the following is true regarding exons in the PNS? I'll give you the question later. The options, but you should know that a Schwann cell wraps its plasma membrane around each individual exon. So you need to know these things for you to be able to give the option, because they can say which of the following is true regarding exons in the PNS. They will give you all these things. They will take one of these and add to it. All right. So you need to know this and you need to know what this. So take time, pause this video and look at it properly, because you cannot afford to fail any question here. In terms of regeneration, you need to know what is the processes for nerve regeneration. First of all, ask yourself, why will a nerve regenerate? Why is regeneration found in PNS instead of in CNS? Think about it. Remember that the PNS has what? Schwann cells, right? And Schwann cells secrete what? Growth factors. Secretes growth factors. These growth factors helps the nerve in the CNS to regrow. CNS does not have Schwann cells, so that is a negative for it. Also, the PNS has something we call neurolemma. This neurolemma provides the pathway, the guidance. But CNS does not have neurolemma, so no guidance. So it's suffering from lack of Schwann cell, no neurolemma. Instead of having a growth factors as Schwann cell produces in the PNS, there is inhibitory signals from the oligodendrocyte. The oligodendrocyte does not want it to grow anything, so it will want to inhibit it. Also in the PNS, we have less scar tissue formation, but in the CNS, glial tissues form scar. And the process of regeneration, the extra to the injury degenerates. Once it degenerates, Schwann cells start forming new tube. Now, exons process from that tube, and from there, growth comes. All right. After that, 1.2 mm of remyelination happens per day from the exon. All right. So it's very, very important. Some questions that they can ask is, in order to regenerate a PNS, exon must have which of the following answer? An exon, which of the following answer? Cell body must be there, because if there's no cell body, everything is dead. And also at least some neurolemma, because neurolemma is what we give it guidance. This is how you should grow. This is how you should grow. All right. Very, very important for you to know this. They may ask you a question about the myelin sheets. A myelin sheet is composed primarily of dash. The answer is lipids and what? Proteins. Lipids and what? Proteins. Very important for you to know. 70 to 80% lipid, 20% to 30% what? Proteins. Right? So that's the thing. And it has white appearance, right? Why? Because myelin itself is, it's a little whitish. So very, very important for you to know this. Now, talking about the structure, myelin sheet is for insulation, right? In between them is what? Nodes of Ranvier. That's gaps. And we have what? Internodes. All right. So look at it. You see this node of Ranvier. All right. This is internode. This is node of Ranvier. And this is what? Nucleus of what? Schwann cell. So this is definitely what? The PN. Of course, you can see the neurolemma here. This is the nucleus. Extremely important that you know this. And what's the function of the myelin? Electrical conduction. So it prevents leakage of iron. Because it prevents leakage of iron, it reduces the energy cost. That's fewer ions to pump. Make sense? Thereby increasing the conduction speed. And this helps to protect the exon, because the exon is not working too hard. So it's protecting the exon. That's what we call saltatory conduction. Jumping. Pew, 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 pew. Very fast. Galloping like a horse. All right? So bear this in mind. This is the myelin sheet. They will actually, at least they will test you on myelin sheet. Now, what are the factors that affect the conduction sheet? Now we have the exon diameter, we have the myelination. These are the two. They will ask you. Look at the sample question down here. Conduction of a nerve impulse would be fastest in which of the following? All right. What are you looking for? The exon that has the bigger diameter and the exon that's myelinated. Simple. If you look at the diameter, the bigger the diameter is faster conduction. That's direct proportionality. All right. Why? Because there's less stress, less distance. It's just like if a lot of people are running, give them a very wide lane to run and everybody will be comfortable. But if you pack them together, what happens? Somebody will hit one another, somebody will fall. That's resistance. All right. So the range is 1 to 20. You understand? In terms of diameter. Why myelination also helps to increase. Myelination is just serving as enzyme, so increase the speed of reaction as enzymes. So this one increases the speed of what? Conduction from 10 to 100 times. All right, and bear in mind, not all nerves are what? Myelinated. Not all what? Nerves are myelinated. So a faster conduction is a conduction that mixes what? Probably the large diameter and also myelination. Wow, very fast. Example, skeletal muscles. They have large fibers and also they are well myelinated. All right. There are others that are small diameter and unmyelinated. They are very, very slow. All right. Now, things like multiple sclerosis is when there's the damage to what? To myelin sheets. When the myelin sheet is having an issue, there's damage to myelination. All right. So it won't be able to conduct faster. Just the same thing with Tax disease. Tax disease is almost the same thing with multiple sclerosis. The only difference is that in multiple sclerosis, the people, they last up to 20 to 30 years before it becomes fatal. All right. That's the range of fatality. But Tazax, the young ones, they die very fast. In 4 years, they are gone. All right. Tazax is usually because of the parts covering the myelinated part. So that part is inhibiting it from increasing the conduction rate. Before you know it, the person starts suffering from eye problem, heart problem, anywhere that has nerves that needs myelination will definitely be affected. All right. So it's very, very fatal. Now, we want to discuss why myelination increases speed. What is the physics behind it? I want you to think of the local train and the express train. All right. If you are in the country where train is being used, there's something called local train. Local train, at each place it stops. At each place it stops. So if you take me 20 or 30 stops to go from Staten Island here to Bronx with local train, with express train, it will take me something like maybe like 7 stops, and I'm in Bronx. So it makes it very fast. That's why you see everybody scrambling, rushing for the express train. It bypasses a lot of stops. So that's exactly what myelination does. All right. First of all, there's electrical insulation. That insulation will make all electricity to pass through. None will leak out to another cell or to another membrane. All right, thereby reducing capacitance. So all currents flows internally instead of what? Crossing membrane. That's the first thing you need to know. That's the electrical insulation. 2, there is node concentration. All right. So the voltage gated channels only add nodes of what? Run there only. So we don't waste channels in the myelin parts. So pew, 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 efficient resource use. So that's node concentration. So we have electrical insulation. We have node what? Concentration. Electrical insulation, node concentration. Now we have circulatory conduction. That's jumping, galloping from here to here. One myelin sheet, it jumps all the myelin sheets, goes to another internode. All right. So it moves very, very fast. So fast step-by-step propagation. Then we have energy efficiency, because if there's no myelin sheet, that means a lot of sodium potassium ions will be needed. Ions will be pumped, right? So it reduces that amount, thereby reducing what? The ATP consumption. Remember, sodium and potassium ion channel pump is an active transport, so it requires ATP. All right, requires ATP. So that's just the thing. Electrical insulation, node concentration, circulatory conduction, and efficiency of energy use. All right. So they may ask you a question like, what would be the best explanation for why myelinated axons conduct signals faster? And the answer is what? Electrical signals spread faster through insulated regions of the axon. Very important that you know this. Now they can ask you a question about the RMP. Which of the following ions has the greatest influence on the resting membrane potential? Now for nerves, we know that the typical resting membrane potential is minus 70 MV, right? Yeah. So the inside is very, very negative. Why is it negative? It's negative because, like 3 sodium is pumped out and 2 sodium is pumped in, meaning that there is more positive that went out than positive that went in, right? So it creates the concentration gradient, all right, making it negative inside. More positive goes out than more negative that's coming in, all right? So the potassium has the greatest influence, all right, because that's what determines whether it is negative or not. Now, when we want to process action potential, what happens? Sodium jumps in and makes it positive. Potassium goes out, makes it negative, and before you know it, action potential is created. Sometimes we have chlorine also coming in. That one causes hyperpolarization, but we'll discuss it later. But when they are talking about what has the greatest influence on the resting membrane potential, always remember what? Potassium. How about the properties of neurons? They can ask you a question. What property of neuron allows them to respond to the changes in the environment? They can ask you other properties. Oh, what property of neuron helps them to produce neurotransmitters? What property of neurons help them to be able to propagate action potentials? We have excitability or irritability, we have conductivity, and we have what? Secretion. These are the 3 major properties. We have other properties like permeability, convergence. All right. Permeability talks about selective membrane channels. Convergence talking about integrating multiple inputs. And when you're talking about excitability, we are talking about the ability to do what? To respond to change, to stimuli. Chemical, mechanical, electrical, any stimuli, it has to respond. All right? So it allows the neurons to sense their environment. And this is what they usually ask you about. Change in membrane potential. You understand? It's always, responded to by excitability. Also, we have conductibility, which is our ability for it to what? Propagate action potentials along its axons. It can be rapid, it can be long distance, it's always very rapid anyway, and it can be long distance communication. So that ability to transmit that information is called conductivity. And secretion, if you check out when action potential gets to that extreme, right? Extreme part of what? Of the axon. What do you think happens there? The action potential causes calcium to enter. Calcium enters, bursts the vesicles, pushes out what? Neurotransmitter. Oh, neurotransmitter. That means neurotransmitter is what? Produced. Neurotransmitter is what? Produced. What produces neurotransmitter? That is what? The nerve, the neuron rather. So it's very, very, very important for you to know that the secretion part is secretes what? Neurotransmitters. What causes that neurotransmitters to be pushed out through exocytosis? We call it what? Calcium influx. Very important that you know this. So when they ask the question about what property of neurons allows them to respond to changes in the environment, that's the excitability. But they can ask this question in different ways. Now, another thing you need to know is action potential and local potential. I mentioned it from the outset of this discussion about local potential that does not pass the axon what? Hillock into the axon. All right. Now, local potential can be graded, they can be decremental, they can be reversible, and they can be summed up. These are also ways where they differ from action potential. What is the meaning of graded? They can vary in magnitude with the stimulus strength. Stronger stimulus will lead to larger potential. Weaker will lead to what? Weaker potential or small potential. Also, local potentials can be decremental. All right. What do we mean? When they keep going, they get tired, they get weaker, and they reduce. Sometimes they disappear. Also, they can be reversible. They can return to RMP without completing. Right? They can keep. They want to get to this place. They go. Boom, they stop. But action potential, if it starts here, it must. As far as action potential has been activated, it's all along law. It must get to this point. And action potential cannot be what? Reversible. It does not reduce. It must get to the extreme part of the axon, cause calcium influx for neurotransmitters to burst and cause action potential in the next neuron. Very important that you know this. Also, action potential is not graded. It doesn't get weaker. So I want you to know that local potential is typically formed where? It's formed in the dendrites and what? Cell body. But axon hillock does not allow it to go. All right? And it also it's usually happens at post-synaptic membranes a lot of times. It can also be caused by ligand-gated, chemically gated channels, neurotransmitter binding, and also mechanically gated channels. Bear in mind this. There are a lot of questions they ask. There are a lot of questions they can ask. They can ask you local potentials are dashed, meaning that they vary in magnitude according to the strength of the stimulus. They are what? Graded. Where do most local potentials form in a neuron? It's usually the dendrite. Axon hillock is just the coordinating center. All right? Local potentials are dashed, whereas action potentials are dashed. You need to know. So these are the things that local potentials are, and I've already discussed the things that action potentials are not using local potential. So you should be able to answer those questions. Very, very important. Now you need to know the three concepts: depolarization, repolarization, and hyperpolarization, and what causes them, because they will ask you questions. Questions like in a neuron, the opening of sodium-gated channels typically leads to dash. Which of the following will cause the plasma membrane to hyperpolarize when it's RMP? What causes hyperpolarization of a neuronal membrane? These questions need to be in your mind. All right. Very, very important that you understand this question, because these are what they are definitely going to test you on. So you need to pay attention. All right. Take your pen and paper and write these things down. Pause me, because I speak very fast. So sometimes you have to pause or slow it down, slow the video down, so that you'll be able to understand me properly. Watch this video as many times as possible. And mind you, you have to subscribe. All right. Subscribe. Put on your notification button. Send this video to your friends as well. All right. The link to your friends so that they can watch it and be able to be prepared for their college exams and for their NCLEX and any exam that you're writing at this level. Depolarization is depolarization becoming positive. That's what depolarization means. Either becoming positive or less what? Negative. Remember, the RMP is -70, right? Yes. So if it gets to 0, that's it's getting more positive. That is what depolarization. All right. What causes depolarization? 3 things. Remember, depolarization is excitation. The depolarization list is excitatory because there's increase in action potential forming. When it goes up, there's increasing action potential forming, all right? Because it gets to threshold, or immediately it gets to that threshold, it forms action potential. So it's excitatory. Three things you have to put it in your head. One, if sodium channels open, what do you think will happen? Sodium will rush inside, because sodium is in ECF, so it will rush inside the ICF, intracellular environment. All right. When it rushes inside the intracellular environment, it leads to positivity. So that's one of the ways that depolarization happens. So one cause of depolarization, sodium channels opening, sodium influx. Second way is by what? Calcium channel entering inside. Remember, calcium is positive. It can actually enter the membrane, right? Then the third thing is what? Remember, being is potassium. Potassium is inside. So if potassium that is inside, that is the positive, if it's the channel opens and potassium start leaving the cell will become positive. These are the three things that can cause positivity. You can't forget it. I need you to understand it. We have repolarization. Repolarization is when it comes down like this. Everything is going back to default setting. What is default setting? RMP. All right. So restoration to negativity, return after the action potential. It's called recovery. So what causes repolarization? Opposite of everything that causes what? Depolarization. It's as simple as that. Sodium channel closing. So sodium is no longer able to come inside. Potassium channel opening. Potassium, it's not living anymore. All right. Calcium channel closing. And calcium is no longer what? Entering inside. So when all these things happen, it leads to what? Repolarization. You have to bear in mind this. Repolarization. So mind you, potassium channel we have to what? Open. So potassium is leaving from inside the cell. So if potassium is leaving, it's going to negative. Calcium has to close. If calcium is closing, it cannot enter anymore. Sodium has to what? Close. So sodium close, calcium close, potassium what? That's the wisdom. We have hyperpolarization, right? What is hyperpolarization? Hyperpolarization is when membrane becomes more negative, all right? It starts going more negative. Minus 70, minus 90, that's more negative. So what happens this time when potassium channels open and potassium starts leaving more? It's minus 70 and potassium is leaving more. It becomes more negative, all right? Or chlorine starts entering, it makes the inside more negative. Closing the sodium channels from entering, more negative. So this is what? Inhibitory, decreases the likelihood of what? Action potential forming. So you need to know depolarization, repolarization, and what? Hyperpolarization. So when you answer this question again, in a neuron, the opening of sodium gates typically leads to what? It leads to what? Depolarization. Which of the following will cause the plasma membrane to hyperpolarize when it's at its what? RMP? It can be, the answer may contain opening of potassium channels, it will be correct. It can contain opening of chloride channels, it can be correct. It can contain closing of sodium channels, it can still be correct. So it can be any of those answers. That's why you need to familiarize yourself very, very well. And the last question there, what causes hyperpolarization of a neuronal what? Membrane? All right. Then you can also choose your answer wisely. Potassium ions leave the cell. Okay. So when you're talking about excitatory and inhibitory potentials, which I've already discussed before, we can call it push or pull. All right. Excitatory, excitation. Inhibitory, inhibit. Excitatory, depolarization. Inhibitory, hyperpolarization. In excitatory, everything because the RMP becomes less negative, more positive. All right. While inhibitory, the RMP becomes more negative. Firing decreases away from pull the membrane away from threshold. But in excitatory, firing increases, push the membrane towards threshold. All right. Now you need to know the mechanism. We've already discussed it before. For excitatory, sodium or calcium channel opening and influxing. All right. For inhibitory, potassium channels open, potassium leaving. It becomes negative, more negative. Chlorine channels open, chlorine enters, more negative. So you need to know which one is entering, which one is what? Exiting. And remember, in the exon hillock, that's where all the summation happens. EPSPs, IPSPs. All right. Everything happens. And if it gets to threshold, it fires. If it doesn't get to threshold, it remains at local potential. So that's decision-making environment. That's exon hillock. You need to understand this. And what's the functional significance? Balance. We want balance. Everything has to be at what? Equilibrium. When there is too much excitation, what does it lead to? Seizure. When there is too much inhibition, what does it lead to? Coma. So they can ask you a question. An inhibitory local potential causes which of the following? Hyperpolarization of the plasma membrane. So you need to understand this extremely well. All right. Now they can ask you what causes rapid depolarization. We've already answered this question. So if you look at this slide, it just gives you a summary of everything. All right. A summary of the entire thing. Opening of sodium channels, what is most likely cause, fast response, inside become positive, sodium rushes in. We discussed this. What doesn't cause rapid depolarization? Opening chloride channels, we cause hyperpolarization, not rapid depolarization. It causes hyperpolarization. Opening potassium channels, hyperpolarization. Closing of sodium channels, depolarization stops. Closing of sodium channels prevents depolarization. You know, like, this is the basic information. So it's just being repeated in different ways, all right, so that it will stick. You need to know what depolarization is, hyperpolarization is, repolarization is, and what causes them, all right? Sodium entry, for example, causes depolarization. Potassium exit causes hyperpolarization or repolarization. Chloride entry causes what? Hyperpolarization. Calcium entry causes what? Depolarization. You know, like things like you need to remember, all right? So the question, ask you a question: which of the following is most likely to cause rapid depolarization? And it's what? Opening of sodium channels. It may not be there. It may be calcium. So you need to check properly your answer. But if sodium is there, please choose sodium. That's what always causes what? Rapid depolarization. Extremely important. Now, this is about action potential, which I will not state for long because we've already discussed it. All or none law. It must get to the threshold before it fires. If not, it's local potential. It's non-decremental. Once it's fired, that's fired. It cannot reduce. All right? It's self-propagating. It travels by itself. It doesn't need you. It doesn't need anybody. It doesn't need its family, its neighbors, its anything. All right? And voltage-gated channels also help because, you know, they are produced along the way. Opening and closing of them is what action potential does, triggers, all right? So the question is, what triggers action potential? Threshold must be reached, usually approximately negative 55 MV, all right? Instead of from negative 70, it becomes negative 55, then action potential, all right? Usually from summation of local potential, as we discussed. What sums these things up? The trigger zone, which is what? Axon hillock. So what initiates action potential? Axon hillock. What causes local potential? The cell body. You need to remember this. It's also, action potential can also be called nerve signal, nerve impulse, traveling wave excitation. Anyone that's called in your exam, please, you should be familiar with that information. They can ask you a question. Local potentials are dash. You're right. Action potentials are dash. That is where they are telling you it must not only be graded. It must not only be all or none law. They can be asking you about decremental and non-decremental, all right? Reversible and non-reversible. You need to know the information by your fingertips, all right? Also, a traveling wave of excitation is known as what? Action potential. So that's why I said you need to be familiar that it's called traveling wave of excitation. You need to be informed that it is called what? Nerve signal or nerve impulse. Anyone they give you, you should be able to answer. Now, if you look at this page properly, I need you to look at the page and be able to understand, you know, each of these things. This is resting state. This is stimulus or local potential development. This is threshold reached at minus 75. This is depolarization. This is repolarization. This is hyperpolarization. All these things are the phases or how action potential forms. All right. Initially, we are at RMP, which is minus 70 MV. All right. Minus 70 MV. Then suddenly there are local potentials. These local potentials are formed in the cell body. They are graded. You know the function. You know the characteristics of local potential. And now, when this local potential now reaches, when they are summed up by the Axon hillock and they reach minus 55 MV, that is what threshold. Immediately it gets there, it is a point of no return. We go nowhere. All right, action potential has to be formed once it gets to this threshold. Okay, now action potential is formed. All right, now this action potential formed. You know, there's something we call, once it gets to this minus 55 MV, the sodium gated channels does what? Open and rush into the cell. Immediately it gets to this minus 55 MV, rush into the cell. Immediately rushes into the cell. It moves the RMP up from minus 50, it moves it up from threshold, that's minus 55, to where? Plus 35. Immediately it moves it up, all right? Then the potassium starts leaving, and potassium starts leaving. What happens? Repolarization happens. Down. Then when repolarization happens, more potassium keep leaving, chlorine even enters, leading to what? Hyperpolarization. That's undershoot. So we have rising phase, falling phase, undershoot. So you need to understand the movement or the phases because they will ask you in the exam. I'll give you a question to that. So all those things I just described initially, both the onrush of sodium, the potassium leaving, going back to negativity, all these things is called what? Voltage changes. All right? Starts at threshold. I told you threshold action potential starts, that's minus 55 MV. And immediately it causes rapid influx of sodium. And once it causes rapid influx of sodium, it rapidly moves towards plus 35 MV, which is the peak. So this change is known as minus 105 MV, all right? Occurs in approximately 1 millisecond. And these are the things that makes it occur, all right? So opening of sodium gated channel leads to what? Depolarization. So that's the question they usually ask. In a membrane, the opening of sodium gated channels typically leads to what? All right. So this is the repolarization phase. They can ask you when the voltage of a plasma membrane shifts from plus 335 MV towards zero. Towards zero, what is it called? The repolarization, because it's going back to negativity. That's the repolarization. All right. So this is sodium channel inactivation. This is potassium channel opening. All these things leads to what? Repolarization. All right. Sodium channel becomes inactive, so sodium is no longer able to enter inside. Potassium channel, open, and potassium starts exiting, leading to what? Negativity. So that's what works here. So this still describes exactly what we talked about, but I want you to look at the sample questions. In an action potential, which event directly follows what? Repolarization. All right. After repolarization, what happened? Hyperrepolarization. Remember, hyperrepolarization is caused by potassium leaving, and also by chlorine entering, and also by shutting of sodium. All those things causes hyperpolarization of the membrane. Now they can ask you what causes hyperpolarization of a neuronal membrane. It depends on the option they give you, but you need to know all the options, which is what we have here, why it occurs. You need to know that. Now there's a question they usually ask. During the absolute refractory period, which of the following is true? They'll give you different options, and most times they'll tell you no stimulus of any strength will trigger a new action potential. Just pick that. Listen, refractory periods are recovery periods. All right. At that moment, anything that goes up must come down. And when it comes down, it's nothing you do it, nothing you trigger it for it to go up. You know what I'm talking about. You understand what I'm talking about. So we have absolute refractory period and relative refractory period. In absolute, no stimulus of any strength can trigger new action potential. In relative, okay, maybe you try it, you put it into different places, you rub it, you touch it, you do a lot of things, and it can possibly trigger what? Action potential, but only with a lot of manipulation and strong word stimulus. All right, so 1 to 2 milliseconds, that's what it takes. All right, it's very few minutes. So immediately it fires, it has to rest before you start engineering to fire again. And it's very important. Why? Because it limits firing frequency and it shows on directional propagation. Imagine if this refractory period is not there. Sometimes the propagation will not even be directional. It will not have one particular location or direction or dimension as well. Also, it prevents tetanus, right? Tetanic contraction. So there are a lot of importance of a refractory period to action potential. Now, what you need to know about communication gap is this is a neuron coming from, this is a particular neuron, this is another neuron, all right? This neuron this is the axon. I told you. Now, I told you initially that axon can match with this neuron. This is presynaptic neuron. This is postsynaptic neuron. Now, that axon this is the axon. This is the leg of the axon. It can pass the information to this other neuron, all right, neuronal dendrites. So if this axon is synapsing with this dendrite, it's known as what? Exodendritic. I mentioned it initially. It can be synapsing with the body. So if this axon is synapsing with this body of this particular neuron, that's postsynaptic neuron, it is what? Exosomatic. And if it can be the axon to axon, all right? So if this axon is synapsing with the axon of this postsynaptic neuron, it is called what? Exoaxonic, all right? The most common is exodendritic. Remember, I told you that most neurons, they receive their information through what? The dendrite. So it makes sense that this one is bringing information, this one is using dendrite, that's the hair, to receive that information. So we have exodendritic, exosomatic, and exoaxonic. Extremely important that you know this. All right. Now, the direction is usually one way. It's unidirectional. It's coming from here, it must go here. All right. Now, you need to understand the presynaptic and postsynaptic neuron. Now, the one that the information is coming from, the action potential is coming from, is what? The presynaptic. The one is going to is what? The postsynaptic. And how does this action potential transfer to this guy here? It's through what? Neurotransmitters. So if they ask you which one produces the neurotransmitter, it's usually the presynaptic. The presynaptic is the one that contains those vesicles, and it has what? Calcium gated channel. So when action potential comes through this presynaptic, it triggers the vesicle and busts them. The vesicle now push out the neurotransmitters through exocytosis. The neurotransmitters now enter this cleft. So the cleft is what? A small gap, just like 20 to 40 nanometers. All right. It's filled with extracellular fluid. All right. So it's just a small gap between the presynaptic and the postsynaptic. All right. Looking at each other like this. So what do you think will happen? The neurotransmitters that came here, we swim across. All right. And they have to match to something here. And what are they matching to here? They are matching to the postsynaptic neurotransmitter receptors. So what produces the neurotransmitter is the presynaptic. And what has the receptors is what? The postsynaptic. I need you to understand. You need to know which one produces the neurotransmitter and which one produces what? Has the what? The receptors. If there's a problem in the receptor, this neuron will not be able to, understand the neurotransmitter. And that's exactly what happens in type 2 DF, because insulin is produced, but there is no receptor. Receptor has, most of them has been destroyed, so insulin will not be able to work. That's why we give medications to increase the sensitivity of the receptors and other things. All right. The same thing with myasthenia gravis. What happens with myasthenia gravis? Oh, the person is producing acetylcholine, or the person has vesicles, and the vesicles has busted through calcium, and it pushed out through exocytosis acetylcholine in the neuromuscular junction between the neuron and the muscle this time around. The acetylcholine has swim in between the synaptic cleft to the receptors, and getting there, the receptors are what? Not there. Like, they are covered. Receptors are covered. Competitive inhibition. So, the acetylcholine is no longer able to trigger the receptors, because that receptors, the acetylcholine serves, the neurotransmitter serves as first messenger. It has to trigger that stuff to cause a conformational change inside the cell. So once the acetylcholine is no longer able to bind to the receptors, there's nothing will happen. That's why myasthenia gravis causes flaccid paralysis. Extremely important. So you need to understand the presynaptic, the synaptic cleft, and what the postsynaptic neuron. Extremely important. A lot of questions is coming here, and also the types of synapses and the direction. Now you need to know the characteristics of what? Of neurotransmitters. All right, because they will ask you all these are typical characteristics of neurotransmitters. Depending on the question, you should be able to answer it properly. I've already discussed it. All right. So these are neurotransmitters synthesized by presynaptic, released in response to stimulation. All right. Action potential comes, calcium busts them open. Bind to what? Specific receptors in the postsynaptic. That's lock and key. You understand? Then after that, it will not cause what? Physiological changes. Immediately binds to the receptors. It leads to what? Calcium channels opening, sodium channels opening in that post-receptor. Calcium channels open here, rather sodium channels. It opens, rushes inside, and this particular cell becomes what? Positive. Potassium leaves, it becomes negative. Chlorine enters, it becomes more negative. Sodium enters again, it becomes positive. Before you know it, action potential is formed, and immediately formed at the exome hillock, it travels and happens to the next neuron. That's what we call neuronal circle. All right. So it's packaged in the vesicles, released by exocytosis, dependent on calcium. So this is just a summary of neurotransmitters, which you must know. So all these things I've been explaining is just the transmission sequences. All right. Vesicles busting, acetylcholine diffusing, joining the receptors, triggers sodium to enter the postsynaptic cell, leading to a postsynaptic potential, is being produced, it leads to excitation, it's always excitatory. All right. There are two types of major receptors. We have the nicotinic and we have the muscarinic. The nicotinic are usually excitatory, the muscarinic can be excitatory or what? Inhibitory. And also clinically, ACH, for example, excites skeletal muscles, but it inhibits what? Cardiac muscles. All right. So they can ask you a question. The cholinergic synapse uses starch as its neurotransmitter. That's acetylcholine. All right. Which of these happens first in a cholinergic synaptic transmission? Cholinergic synaptic transmission. What happens? That is when acetylcholine is released from the vesicles through what? Exocytosis. So this order is what you need to know. All right. For the adrenergic, they are slower, more amplified. They use an epinephrine, not epinephrine, as their neurotransmitters. All right. They use epinephrine. They are very related to what? Adrenaline. They are slower, but they are more versatile than what? The cholinergic system. The first one we discussed initially is the cholinergic system. All right. In comparison to the cholinergic system, the adrenergic system is always slower on onset. They have a longer duration. That's why you can keep running if snake is chasing you. All right. And you'll not get tired. They're more diverse, and they amplify a lot. They make you act stronger than you can. I see a story of a woman when the vehicle fell on the child. The woman lifted the vehicle up. On a normal, that woman will not be able to lift that vehicle, possibly. All right, but because of that adrenergic propagation, she got that strength to be able to do it. That's amplifying her normal strength. All right, very, very important. There are types of adrenergic receptors. We have the alpha, we have A1, A2, we have the beta, A, B2, B1, B2, and B3. All right. All of them, they have their own places that they are found, and they have specific thing or specific way they work. Now, for them, their transmission sequence is that G protein dissociates from the NE receptor. So the first messenger is what? Neurotransmitter. It's lapped on the receptor, and immediately lapped on the receptor, what happens inside? G protein dissociates, and G protein binds to adenylyl cyclase. Adenylyl cyclase is now being what? Activated. It activates what? ATP, converts ATP to what? CAMP, and CAMP is what? The second messenger. It is that second messenger inside that now what? Starts causing conformational changes, start making everything active. So the difference between this and that is that the other one, immediately for the cholinergic system, what happens there? As you mentioned initially, once the acetylcholine diffuses, goes to the receptor on the postsynaptic membrane, it leads to what? Sodium entering. And sodium entering starts the entire increase, it causes positivity in the ICF, and everything leads to what? Excitation. That's like cholinergic system are usually what? Excitatory. Why, if you accept in the hearts, all right? Now, why, if you look at the adrenergic system, it can be excitatory and what? It can be inhibitory, depends on where it's found. And this is how it works. This is how it works. If you have not seen my video on muscular tissue, please go and look at it, because I describe this pathway. When G protein moves, I called the receptor a bouncer. Somebody came and gave the information to a bouncer. That's the first messenger. Gave the information to the bouncer. All right? And that is when epinephrine lapped on the receptor. What did the bouncer do? The bouncer removes the shoe and gave to the second bouncer. The second bouncer is what? Called adenylyl cyclase. Remember, adenylyl cyclase is still in the plasma membrane. That shoe he removed is just like the G protein. So as he removed the shoe, what does adenylyl cyclase do? Adenylyl cyclase now called ATP and told ATP to change his clothes. And ATP changes clothes and become what? CAMP. That CAMP is now the one that is now instructed, oh yeah, go and take care of all the events that has been instructed to us by the first messenger, which is the NE, because NE is not able to enter the cell. So this is the sequence. That's the transmission sequence. All right? If you can remember this story, you are good to go, because they will ask you, which of these happens first in the adrenergic transmission? And that is what? G protein dissociation from the NE receptor. So NE, norepinephrine, lapped on the bouncer. What does the bouncer do? The bouncer removes the shoe and gave to the other bouncer. That removing shoe, that shoe is the G protein, right? That dissociation is the first thing that happens before adenylyl cyclase triggers action ATP to produce what? To become a CAMP. All right? Very important. Now, they can ask you a question about termination of the entire thing. Which of these does not contribute to the cessation of signal in a synaptic transmission? All right? So you look at that question very well. Why is this thing important? It's important to avoid overstimulation, because if you allow overstimulation, that's when we have what? Spastic paralysis, all right, or seizure. So, and also so that we can allow new signal to be detected. You understand? If there is dysfunction, it becomes excessive stimulation, as I mentioned. All right. Like when you have tetanus, a drug targets this a lot. Drugs target this a lot. Drugs like what? Like we have the reuptake inhibitors, like the antidepressants. We have the enzyme inhibitors, like MOA inhibitors. All right, and also the enhancement drugs that we take, prolonged enhancements. All right, like the one that we use for myasthenia gravis. We want it to enhance the amount of acetylcholine that remains in the synaptic cleft. So there are 4 methods for stopping. 1, neurotransmitters are no longer released. All right, there's no more exocytosis. Calcium is no longer entering because action potential is no longer coming to the knob. All right, so calcium stops. So vesicles are no longer busted. That's 1. Neurotransmitters are no longer released. 2, neurotransmitters escapes from synapse. All right, they diffuse back or go away into the ECF. So that's the point. All right, that's dilution or physical removal. We can do it. That's why they do surgery sometimes. Now, the third one is exon terminal reabsorption or neurotransmitter. We call it what? Reuptake. There's endocytosis. Remember, exocytosis pulls it out. Endocytosis takes it back. All right, everything recycles. And drugs like selective SSRIs, all right, SSRIs and cocaine, they do this. They help to reuptake. All right. That's why you have this very calming effect. Oh, you are relaxed, right? Now we have enzymes that break down neurotransmitters, like what? Acetylcholine break down neurotransmitters to acetyl and choline and takes it back into the vesicle. All right. So examples of these MOAs, monoamine oxidases, they break down monoamines. That's why we give monoamine inhibitors to inhibit that breakdown of monoamines. All right. All right. So these are major neurotransmitter classes. I'm not going to discuss them. We have the acetylcholine, we have the monoamines, we have the neuropeptides, we have the purines, we have the gases, we have the amino acids. You need to be familiar with these things. Pause this screen, please, and look at these things because you'll be tested. Purines like ATP, adenosine, they are actually neurotransmitters. Gases like nitric acid, CO2, CO, they are neurotransmitters, but they are not stored in vesicles. All right? They can ask you which ones are not stored, which of the following neurotransmitters is not stored in vesicles. We have amino acids, all right? Usually the glutamates. Glutamates are very, very, very excitatory. Most common in the CNS. We have the GABA, which is inhibitory, most common in the brain. We have the glycine, which is inhibitory, most common in where? In the spinal cord. And that is what tetanus targets. Tetanus will release something that inhibits the glycine in the spinal cord. It will inhibit it. And the glycine is for inhibition. Inhibitory effect. So if what causes inhibition is inhibited, it will lead to what? Excitation. That's why we have lockjaw in tetanus. Lockjaw in tetanus. All right? Then we have aspartate as well. Then we have neuroleptides like endorphins and substance P. All right. Then all of us know acetylcholine. Know that acetylcholine is degraded by what? Acetylcholinesterase. And it is very important in motor control, very important in memory, very important in attention. Very important. Why the monoamines, all right, like dopamine, norepinephrine, epinephrine, all of them, serotonin, histamine, they are for mood, arousal, reward, and also for movement. That's why Parkinson, when there is no dopamine in the substantia nigra, it leads to uncontrolled movements. All right. Okay. We also have glutamate, the excitatory ones, the major excitatory ones. We have glutamate, we have acetylcholine. But mind you, that acetylcholine, it depends on which receptor. We have nicotinic and we have muscarinic. All right. So sometimes it's inhibitory. It depends on where it is. If it's in the heart, it is inhibitory. But if it is skeletal muscle, it's excitatory. You have to understand that. All right. Then we have the norepinephrine, which can also be excitatory and inhibitory, depending. That's why it can be very sympathetic. And remember, sympathetic in the urinary system, sympathetic, it causes inhibition. In the digestive system, sympathetic causes inhibition. All right. Then we have the glutamate. It's the most common excitatory neurotransmitter. So if I want excitation, I trigger glutamate. We've already discussed mechanisms of action, so I'm not going to talk about it. Then we have clinical relevance. All right. Too much excitation causes seizures. Just mind you. So stimulants increases excitatory transmissions. So we want balance. They can ask the question, which neurotransmitter excites skeletal muscle and inhibits cardiac muscle? Which one? Acetylcholine. You remember what I told you? Nicotinic and muscarinic. Okay. We also have GABA. That's major inhibitory, GABA, glycine, and some monoamines. All right. So you look at it, they can ask you what is the most common inhibitory neurotransmitter in the brain. It is always GABA, please. GABA, that's gamma amino butyric acid. That's what we call GABA. All right. And they work with opening chlorine channels, opening potassium channels to cause hyperpolarization. You already know that. And why it's clinically important is because drugs like benzodiazepine, they enhance what? GABA. Anxiety medication. So somebody has anxiety, just enhance the GABA. Why? GABA is inhibitory, so it's calming. I just give you a thing, benzodiazepine, so increase your GABA level, and it will calm you. Also, we use it to calm somebody during seizure. All right, that's because it's inhibitory. And when you give alcohol, that's why you need to know that alcohol enhances GABA. All right. That's why somebody takes alcohol, the person is calm. You understand? So, but too much inhibition, when you take alcohol too much, it leads to sedation, it can even lead to coma. All right. So it's very, very important that you know these things. All right. These are the functional roles. Now think about dopamine as well. I've mentioned it initially. Dopamine is a monoamine neurotransmitter, catecholamine. It's synthesized by tyrosine. Very, very important. What's the function? Motor control in the basal ganglia, reward and motivation, attention and learning, inhibits excessive motor activity. If there's a problem with dopamine, what happens? All these things are affected. All right. All of them are affected. Parkinson's disease is an example of dysfunction of dopamine. All right. Why? Because a lot of dopamine producing neurons are destroyed. And remember, that's why it's called progressive disease. It keeps continuing. All right. Usually the substantia nigra degenerates because that's where you see a lot of them. What does it lead to? Tremor, rigidity, bradykinesia, postural disability. So dopamine normally acts as what? Inhibitory. It prevents excessive motor activity. So when excessive that inhibitory is reduced, it leads to what? Excited, uncontrolled activity. All right. Periodically, they can ask you a question. Parkinson's disease involves neurons that secrete which neurotransmitter? Dopamine. Some antidepressants drugs inhibit MAO. What is the function of the enzyme? It breaks down what? Amine. Simple. So monoamine oxidase breaks down what? Monoamine. So we use them as antidepressants many times. All right. Also to treat Parkinson. So that leads us to excitatory postsynaptic potential, that EPSPs. All right. This is the definition. The mechanism is open sodium and calcium. We've already discussed it. Characteristics is graded, summation, possible, decremental, shortening, just like local potential. All right. Grade potential relationship. All right. These are the things that causes IPSP to happen and EPSP to happen. All right. The question can be the opening of dash gates produce dash. Simple. What are they asking you? Look for it. Well, look in the option. It may not be calcium. It may be sodium. All right. So if the answer is calcium, opening of calcium, what will it lead to? It should be producing what? EPSP, not IPSP. You should understand that. All right. But opening of potassium, opening of chlorine will lead to what? IPSP. It's as simple as that. So this is IPSP as I was talking about. They will ask you a question: inhibitory postsynaptic potential associated with which of the following? All right. Inhibitory postsynaptic potential is always what? Hyperpolarization of the cell membrane. Simple. What leads to it? Chlorine, even potassium. So remember that. But chlorine what? In, potassium out leads to negativity. You have to know which one is going in, which one is coming out, so that they will not what? Confuse you. Now you have temporal summation and spatial summation. Temporal is usually when there is stimulation from one synapse. Spatial is when there is summation from what? Multiple synapses. All these things happens where? In the axon hillock. Remember, all right? So that spatial can involve hundreds of synapses, all right? So the decision, if EPSP, all right, is greater than threshold, what will it lead to? Action potential. If it's less than threshold, no potential. Simple. This has functional significance, even clinical correlation, all right? So I need you to look up this later. I'll give you questions that will treat this. You need to understand the difference between nervous system and endocrine system. I've already mentioned from the beginning what each does. Now, this is a summary of the entire discussion, all right? Signal, how does it flow? Stimulus to receptor to sensory neuron to CNS, it returns through the motor neuron, effector, and the response happens. All right, the one going to CNS is what? The efferent. The one coming back from CNS is what? Efferent. All right, the one going to the CNS is sensory. The one coming back from CNS is what? Motor. All right, and I also told you that the one that is coming back, that is motor, can either be coming to the skeletal muscle, coming to the smooth and the cardiac. Even the one that is going can either be going from the skeletal muscle or the smooth and the cardiac. If it's going from the skeletal, that is what? If it is from anything, either it's going from the skeletal or coming to the skeletal, it is what? It is what? I want you to put the answer in the comment section. It's somatic. All right. If it is going to the smooth muscle or the cardiac muscle, it is, or anything that is not skeletal, it is what? Visceral. Now, the one coming back from the brain to the viscerals, all right, that's the visceral motor, can be divided into two. We have the sympathetic and non-sympathetic, or parasympathetic rather, sympathetic and parasympathetic. So you need to be able to place them together. Then we discuss the neuron types, functional classification. We talk about the sensory, the motor, the interneuron, and we talk about the structural classification. That's the multipolar, bipolar, unipolar, anasomic. We talked about what cell body is. That's my face. The dendrite is the input. Cell body is the metabolic center, while the axon is the output. And what coordinates the entire thing? That is the hillocks. All right. Then we talked about the glial cells, the supporting cells. You understand, in the CNS, we have 4 of them. We have the oligodendrocytes that produce myelin. We have the astrocytes that helps in BBB, blood-brain barrier. We have the microglia, that is the white blood cell, the mop up, the genitals, the lymphocyte, anything you want to say about it. Then we have what? The ependymal cells that helps with the correct plexus to form what? To form the CSF, all right, and redistribute it. Now, in the PNS, we have what? We have the Schwann cell that produces myelin. And we have the satellite cells that oversees everything, and also to protect the electrical activity in the cell body, in the PNS. And we also discussed that PNS has neurolemma and it has Schwann cells, and because of this, it can regenerate. Unlike the CNS, that it's not easily regeneratable. Why? Because the oligodendrocyte tries to inhibit any regeneration, but the Schwann cell encourages regeneration. Now we discussed about the electrical signals, all right? RMP minus 70, and what has the greatest influence on RMP? That is potassium. We talked about the local potentials, graded the characteristics of local potentials, and differentiate it from the action potential. Local potential is graded, action potential is not. Local potential is decremental, action potential is not. Local potential is reversible, action potential is not. Local potential is summative, action potential is not. Instead, action potential has all or none law, non-decremental, self-propagating. And myelination helps, the diameter helps to increase what? Speed of what? Conduction. For myelination, we call it what? Saltatory conduction. And we also happens to discuss the sequence of action potential, all right, which is local potential, which reaches threshold minus 55. Then if it reaches threshold, it leads to what? Depolarization. Depolarization, sodium enters, peaks at plus 35 MV. Pium. Then repolarization. Pium. At the repolarization, potassium is going out, returning back to negativity. And after that, more potassium keep going out and negativity. That's hyperpolarization. And at the end of the day, it returns back to what? RN. Now we discussed the refractory period. The refractory period, we have the absolute, we have the relative. Absolute is where when there is no stimulus can make it work. Actually, potential has gone. All right. No stimulus can make it what? Go again. It has to rest. All right. Sodium has been inactivated. But relative, with the strong stimulus, you can make it, you can activate it again if it's relative. All right. Now we have synaptic transmission. We have the cholinergic, we have the adrenergic. Cholinergic is fast, direct, ligand-gated. Adrenergic is slow, indirect, using G protein, CNS, which we discussed. All right. Termination is also as a result of when they stop releasing neurotransmitter, diffusion of neurotransmitter out of the cleft, reuptake or enzymatic breakdown, like acetylcholinesterase, which breaks down acetylcholine into choline and acetyl and back through endocytosis into the vesicles in the presynaptic nerve. Now, we also talked about neurotransmitters. We have excitatory, we have inhibitory. All right. The excitatory, the major excitatory is glutamate. ACH is also excitatory, but only in the skeletal muscle. That's acetylcholine. Then we have the inhibitory, which is the GABA, only in the brain. We have the glycine in the spinal cord. We have the dopamine for motor. All right, in the basal ganglia. All right, that we say ACH excites skeletal muscle, inhibits cardiac muscle. We already discussed. We also talked about postsynaptic potentials, EPSPs and IPSPs. EPSPs causes depolarization using calcium, using sodium, excitatory. IPS, hyperpolarization, potassium out, chlorine in, inhibitory. All right, this integration happens at the halo. All right, then we also compare the system comparison, which is nervous system and endocrine system, because they are very related. They help for internal coordination. So the nervous system, we say they use what? Neurotransmitters in synaptic clefts. All right, and they are very fast and very specific. While the endocrine system uses hormone, pour it into blood before it travels fast. So it travels what? Slow, but it is more widespread. Clinical relevance, talk about Parkinsonism, myasthenia gravis, multiple sclerosis, Alzheimer, epilepsy. All these things are the things that we understand from nervous tissue, because a problem to nervous tissue causes all these issues. All right, I'm not discussing pathology. I'll make a video for it, but just know that Parkinson's disease is as a result of loss of dopamine neurons in the basal ganglia, specifically in the substantia nigra. All right, so motor problems becomes huge. All right, start moving uncontrollably. Then we have demyelination, because the receptors are blocked. That's multiple sclerosis. All right. Then we have epilepsy, excessive excitation or loss of inhibition, leading to seizure. Then we have myasthenia gravis, which is blocking of the ACH receptors, just like multiple sclerosis, leading to flaccidity. Also, we have Alzheimer, which is loss of what? ACH neurons. ACH acetylcholine helps what? In memory. So if the neurons are lost, acetylcholine will no longer be able to function well, leading to memory problems. And the drugs we target are drugs like what? That are used are ion channel blockers. All right, like local anesthetics that block sodium, that's sodium channel blockers. We also have neurotransmitter synthesis, like L-DOPA, increases DOPA in the brain. All right, because dopamine is a problem. We have the selective serotonin reuptake inhibitors, like SSRIs, that helps for depression. Even cocaine that helps to reward. Then we have the ones that causes degradation. All right, like the MOA inhibitors. Because they degrade MOAs, all right? MOAs are monoamine oxidases, all right? So this helps to solve the problem of depression and Parkinson. Also, we have the beta blockers. This one blocks the receptors. We use it for the heart, all right? And also to slow down the heart, and also benzodiazepines to slow down any issues somebody's having. That's for anxiety. Now, I'm going to ask you a few questions. Now, let me ask you a question. In which part of the neuron is the postsynaptic membrane usually found? Axon terminal, axon, dendrites, axon collateral is usually the dendrites. All right. There are a lot of questions that you can be asked, which I've already treated, but I still just want to talk about some of those questions. All right. If you have the patience to listen to me, they can tell you most metabolic and regulatory functions in a neuron happen where. They can tell you the axon, the cell body, the dendrites, the helix, the Schwann cell. And what is the answer? Put it in the comment section. It's usually what? The dendrites. Also, they can ask you a question: what is the term, the nerve fiber? What does it refer to? It refers to axon. What is the most common type of neuron? It is a multipolar neuron. About 90% of the neurons in the nervous system are interneurons. Remember, I told you it's the one with the highest concentration. They can tell you that some dash neurons are specialized to detect stimuli, whereas dash neurons send signals to the effectors of the nervous system. That's efferent and what? efferent. So these informations you need to know. I've already asked all these questions before. I'm just testing if you can remember. Nerves are dash of the nervous system. Are they organs? Are they tissues? Are they organelles? Are they cells? Nerves are what? Organs. They will ask you these kind of questions. All right. What is another name of the autonomic nervous system? It is called what? Visceral motor division. Autonomic, visceral. Remember I told you that there are two classifications. All right. The one that is going to the cell, the one that is going to the smooth muscle, and the one going to the smooth muscle and what? And cardiac muscle is known as what? Visceral. And if they are coming from the CNS, it's known as what? Motor division. So visceral motor division. Visceral motor division is a type of autonomic what? Nervous system. All right. Even visceral sensory is also a type, but the major answer there is visceral motor division. There's a reason which I will explain when we are talking in type 2 or the neuroanatomy 2. All right. Now they can ask you which one is effector. Okay, let me ask you this question. Which of the following is effectors? Receptors, stimuli, reflexes, glands, sense organs. I want you to put the answer in the comment section and I will tell you if you are right or wrong. Then the dash division tends to prepare the body for action. It's always sympathetic. Remember, sympathetic and parasympathetic is a division of what? The visceral motor. So remember it. It's a division of what? The visceral what? Motor. The dash division carries signals to the smooth muscle in the large intestine. It's always visceral motor. You cannot fail this question. All these questions I've already asked them. Which of the following has the greatest influence on the resting membrane potential? It's potassium. We discussed it. I hope you didn't forget. All right. Where do most local potentials form in the neuron? Where do they form in the neuron? Local potentials are usually formed where? In the dendrites. Very, very important. In a neuron, the opening of the sodium gates typically leads to what? Depolarization of the plasma membrane. We discussed this. Which of the following will cause the plasma membrane to hyperpolarize when at its RMP? We have an inflow of sodium, an inflow of potassium, an inflow of calcium, an inflow of chlorine, an outflow of chloride. Put the answer in the comment section. Tell me the particular question and write the answer so that I'll be able to know what you're answering, and I'll tell you if you're right or wrong. All right? Local potentials are dash, meaning they vary in magnitude according to the strength of the stimulus. Stable, graded, self-propagating, excitatory, inhibitory. What's the answer? Put it in the comment section below. Tell me, write the question and write the answer, and I'll tell you if you're right or wrong. An inhibitory local potential causes which of the following? A. Depolarization of the plasma membrane. B. Hyperpolarization of the plasma membrane. C. Repolarization of the plasma membrane. D. Neutralization of the plasma membrane. And E. Drifting of the membrane potential toward the resting membrane potential. What's the answer? The answer is hyperpolarization of the plasma membrane. Please remember this. Which of the following is most likely to cause a rapid depolarization? A. Opening of the chloride channels, opening of the sodium channels, opening of the potassium channels, inactivation of the sodium channels, closing of the sodium channels. The answer is what? Opening of the sodium channels. I've discussed it before a lot of times. I keep mentioning it. A neuron receives stimulus. Which of the following events happen next? All right. A local potential develops, a threshold is reached, hyperpolarization of the membrane, repolarization of the membrane, a resting membrane potential is established. The answer is always what? A local potential what? Develops. So you need to know which one is the right answer. So we've already discussed a lot of questions. I need you to take your time. You see the questions on my slides? Look at those questions. Look at those questions. Know why the answer is correct. Look at the slides properly. Listen to what I explained about each slide. Pause the video and understand it properly. By so doing, you'll be well equipped for this part. I will be dropping the second neuro video very soon. All right. So I need you to subscribe, make comments, like, and click your notification bell so that immediately I drop it, you'll be able to receive this information. All right. Thank you once again for being with me throughout this discourse. I appreciate it. I wish you success in your exam. And mind you, this exam is not only for the college. This presentation is not only for the college exams. It's for your AP 1 and 2 for college, and also it's for your board exams. All right, the NCLEX, USMLE, and others. For you to be able to understand different concepts, you need to pay attention to this video. Take your pen and paper, jot down, write down the questions, think about them. The questions can come in different forms. This is just an example of the questions. All right, and mind you, this does not substitute for your slide, does not substitute for your lecture. Thank you so much once again for coming to my channel. I'll see you later.